This lecture explains gene dosage compensation through X chromosome inactivation in mammals. Chromosomal composition of human somatic cells. This figure shows the chromosomal composition of human somatic cells. Human somatic cells are 2n. Diploid, so there are pairs of autosomes numbered, from 1 to 22, in order of decreasing size. As for sex chromosomes, females have 2 X chromosomes, while males have 1 X chromosome, and 1 Y chromosome. Comparison of X chromosome and Y chromosome. While the X chromosome is composed of approximately 165 million base pairs, the Y chromosome consists of about 60 million base pairs. Consequently, the size of the Y chromosome is less than 1/3 of the X chromosome. The X chromosome encodes 1098 genes, while the Y chromosome encodes only 78 genes. The Y chromosome contains genes related to sperm formation and male hormone production. These genes are not present on the X chromosome. Although the X and Y chromosomes differ greatly in size and internal base sequence, they pair up during meiosis, like autosomal chromosome pairs. The pairing of X and Y chromosomes is possible due to the presence of pseudo-autosomal regions, par1 and par2, at the ends of the X and Y sex chromosomes. These regions contain highly conserved sequences, shared by both chromosomes. X chromosome inactivation. In 1949, Professor Barr discovered that, when staining chromosomes in female somatic cells, a consistently stained dot could be observed, which he named the Barr body. Barr bodies are not observed in male cells. It has been found that the Barr body is one of the 2 X chromosomes, in a condensed state. In the case of XXX cells, 2 Barr bodies are observed, and in XXXX cells, 3 Barr bodies are observed. The Barr body test was one of the sex verification methods used for Olympic athletes from the 1960s to the 1990s. Abnormalities in sex chromosome composition, and their effects. When a fertilized egg has an abnormal number of X chromosomes, mild abnormalities may occur, but no abnormalities that significantly interfere with daily life appear. Women with only 1 X chromosome exist, at a rate of 1 in 1,000 to 2,000 people. In this case, there are no major problems in normal life, but there is a tendency for short stature. Women with 3X also exist at a rate of about 1 in 1,000 people. In this case as well, there are almost no physical characteristics. Men with an XXY also exist at a rate of about 1 in 1,000 people. In this case too, there are no problems in daily life. On the other hand, fertilized eggs with only Y or YY result in spontaneous miscarriage very early in development, so such humans do not exist. This is because the X contains many genes, necessary not only for sex determination, but also for life maintenance. So, chromosomes with only autosomes in Y do not have enough genetic information to form an individual. Conversely, there are no genes necessary for life maintenance on the Y chromosome. Therefore, individuals with XO or XX can survive. From these facts, genes on the Y chromosome are needed to form an individual with male characteristics, based on a female. In somatic cells, one X is in a state where genes can be transcribed, while the other X is highly condensed and inactivated, which is the normal state. For this reason, XO individuals do not have significant differences from XX individuals. Also, in XXX individuals, there is a mechanism by which 2 of the 3 X chromosomes are inactivated, so no major abnormalities are observed in XXX individuals. Effects of autosomal trisomy. Next, I will explain the effects of having 3 copies of specific autosomes. Trisomy. Autosomes are numbered, in order of decreasing size. When large autosomes, such as chromosomes, 1 to 6, become trisomy, it is known to result in miscarriage. On the other hand, when smaller chromosomes become trisomy, various syndromes develop, but miscarriage does not occur. Because smaller chromosomes encode a limited number of genes. In the case of trisomy, there is no deficiency in the genetic information. Nevertheless, trisomy causes various severe abnormalities, because the increased metabolites from specific genes that can have harmful effects on the cell. Unlike the X chromosome, autosomes do not have a mechanism to detect the number of chromosomes and inactivate excess ones. Mary Lyon's hypothesis. In 1961, Mary Lyon proposed a hypothesis regarding the Barr body, as illustrated in the diagram. This hypothesis was later confirmed to be correct through molecular biological research. First, in female somatic cells, one of the 2 X chromosomes is inactivated. Second, inactivation occurs very early in development. Third, the inactivation is random. Fourth, the inactivated X chromosome is stably maintained through subsequent cell divisions. Random inactivation of X chromosome. In XX fertilized eggs, the inactivation of either the maternally derived X or the paternally derived X occurs randomly. The determination of which X will be inactivated takes place during the blastocyst stage, approximately 5 days after fertilization. Once an X chromosome is inactivated, this inactivation is maintained through subsequent somatic cell divisions. As an exception, in the germ cells of the fetus, the condensation of the inactivated X chromosome is reversed. Consequently, in the next generation, both sperm and eggs contain activated X chromosomes. X chromosome chimera in adult individuals. As a result of random X chromosome inactivation selected during the blastocyst stage, being maintained throughout subsequent development, adult female bodies exhibit a patchwork distribution regarding X chromosome selection. Gene expression related to calico cat coat color. In cats, genes determining fur color are located on the X chromosome. If the dominant gene O is present, black pigment is deposited, while in the case of the recessive gene O, brown pigment is deposited. On the other hand, in regions where the autosomal gene S is expressed, pigment deposition is inhibited, resulting in white fur, regardless of the genes, on the X chromosome. Mechanisms of X chromosome dosage compensation in various organisms. In species with XY sex chromosomes, the mechanisms of sex chromosome dosage compensation vary among different organisms. In mammals, one of the X chromosomes is completely inactivated, ensuring that the expression level of X in cells with XY is equal to the transcription level from X in cells with XX. In contrast, in Drosophila, fruit flies, the expression level of X is doubled, only in XY males, compensating to match the expression level in XX female cells. In nematodes such as C. elegans, XX individuals produce both sperm and eggs, while XO individuals are males. In cells with XX, the transcription level of both X chromosomes is halved, to match the transcription level in XO cells. Genes expressed on the inactivated X chromosome. The pseudo-autosomal regions, PAR1 and PAR2, are located at both ends of the X and Y sex chromosomes, AAA, and these regions are used for XY chromosome pairing during meiosis, as previously introduced. Genes in these regions escape inactivation, even on the inactivated X chromosome, and are not subject to expression suppression. The PAR regions are conserved on both X and Y chromosomes, and encode common genes. So whether X10 or 10Y, genes in these regions are expressed from two copies, similar to autosomal genes. In humans, it has been found that a considerable number of genes outside the PAR regions on the X chromosome also escape inactivation. About 30% of all X chromosome genes are expressed. For such genes, two copies are expressed in XX individuals, while only one copy is expressed in XY individuals. This is the reason for the mild abnormalities observed in individuals with XO, Turner syndrome, or XXY, Klinefelter syndrome. Discovery of the X inactivation center. It was found, when a specific region on the X chromosome is deleted, such a X chromosome does not undergo inactivation. So, this region was named X inactivation center. This region consists of approximately 1,000 kilobits. Furthermore, it was discovered, when this region is transplanted to an autosome, that autosome becomes inactivated. Additionally, an about 17 kilobits long non-coding RNA was detected, expressed only from the XIC region, on the inactive X chromosome. The gene for this RNA was named X inactive specific transcript, XIST. The XIST gene was found to have little to no expression on the active X chromosome. X chromosome inactivation by XIST RNA attachment. Subsequent studies have shown that the non-coding long RNA, which is a transcript of XIST, attaches to almost the entire X chromosome, from which it is transcribed, but excepting for the regions such as pseudo-autosomal regions. Two non-coding long RNAs encoded within the XIC, XIST and TSIX. Subsequent research has revealed that, in addition to the XIST non-coding long RNA gene, the XIC region contains a gene that transcribes TSIX non-coding long RNA as an antisense transcript from its complementary strand. Moreover, it is discovered that TSIX RNA functions to suppress the expression of XIST RNA. Determination of the inactive X chromosome through XIC region pairing. In undifferentiated somatic cells, both XIST and TSIX are expressed at low levels, on both X chromosomes. Eventually, a phenomenon of X chromosome pairing is observed. During this time, the XIC region is also observed to pair for about 40 minutes. It has been discovered that, through this transient pairing of the XIC region, the inactivation of one of the two X chromosomes is randomly determined. Immediately after pairing, TSIX RNA expression is transiently suppressed on one of the X chromosomes. Due to the decrease in this inhibitory RNA, XIST expression is activated on this X chromosome. The transcribed XIST RNA covers the surface of that X chromosome, inducing the X chromosome inactivation. On the other hand, on the X chromosome, where TSIX RNA expression levels did not change, XIST expression remains at a low level, so inactivation does not occur. Determination of the inactive X chromosome through unequal distribution of XIC-associated proteins. After transient pairing of X chromosomes, then, the XICs separate. This pairing and separation results in an unequal distribution of proteins involved in chromatin structure regulation that were once evenly attached to the XIC regions. This determines which X chromosome will be inactivated. Among the chromatin structure regulation proteins, CTCF has garnered particular attention. X chromosome inactivation involving histone modifications and DNA methylation. On the inactivated X chromosome, in addition to being covered by XISD RNA, there are several characteristics. A higher frequency of repressive histone modifications. A lower frequency of histone acetylation. A higher rate of DNA methylation. Through these epigenetic chromosome modifications, the condensation of the inactivated X chromosome is stably maintained. Process how the inactive X chromosome is determined. First, in undifferentiated cells, both X chromosomes have low-level expression of XIST and TSIX genes. Both X chromosomes are not condensed at this stage. Second, the mobility of X chromosomes within the cell becomes more active, promoting collisions between the X chromosomes. Third, a brief pairing occurs between the XIC regions of the two X chromosomes. Fourth, the XIC regions separate, leading to an uneven distribution of CTCF and transcription factors. As a result, TSIX RNA expression is suppressed on one X chromosome, which in turn activates XISD RNA expression, leading to the transcription of large amounts of XIST RNA. Fifth, the X chromosome expressing large amounts of XIST RNA becomes covered by XIST RNA, initiating high-level chromosome condensation. The content of this video can be viewed in text and images on the home page site, biology website, Life Science Lectures for you. The site address is given in the description field of this video.
I now conclude this lecture.
If you have not found it yet, keep looking.